Hello, my name is Michaela Barnes, and today I'm going to teach you about the basics of fluorescence microscopy. So, starting with the basics, many of you have probably seen a microscope at some point. These instruments use visible light to magnify tiny structures like the cells that make up our bodies. While these tools are helpful for many labs and science classes, there is a limit as to what they can show us. The picture of the microscope on the right shows all of the different parts. The stage right here is where the sample or what you're trying to look at is placed, and these little clips are what secure it. The two lenses at the top are the eyepieces and allow you to see the magnified version of your sample. The revolving nose piece down here changes the magnification, making it more or less zoomed in. And then the light source goes through the condenser to create the magnification of the microscope. Sometimes, though, it's difficult to differentiate between cells under the microscope. It can almost be compared to trying to see individual stars in broad daylight. The overexposure of the light used can sometimes make it hard to identify specific structures, which is why it's a good thing there's more than one technique. And over here we see platelet cells that are located in our blood, and we see how it can kind of get hard to differentiate between the cells. So, fluorescence microscopy can almost be thought of as turning off the sun and lighting up the stars with a special glowing color. Scientists use fluorescent dyes, which are like a glowing highlighter color, that attach to specific parts of target structures. When these dyes are hit with a certain type of light that the fluorescence microscope uses, the dyes illuminate in bright colors. This technique makes what was invisible or hard to see visible in stark contrast, as we see in this picture to the left. So the basic steps to use the microscope begin with setting it up. Start by removing the protective cover and make sure the microscope is set on low power before plugging it in and switching it on. Then turn on the lamp and wait for about 15 minutes to ensure that illumination of your sample will be strong. Make sure you avoid looking at the lamp directly, as it releases very potent UV radiation, similarly to how you don't want to look directly at the sun. Switch on the motorized focus box on the microscope. Step 2. Place and focus the sample. Place the slide, the piece of glass containing the sample, onto the stage and secure it with the clips. Using the knobs on the side of the microscope as you peer through your eyepiece, you can focus the sample in your field of vision. Step 3. Capture the images. A camera can be attached to the eyepiece to take a picture of the sample and upload it to a computer or device for viewing. Step 4. Power down the microscope. After the microscope has been on for at least 30 minutes, you can safely switch it off to ensure the lamp will be cooled properly. Fifth and final step, replace the protective cover and store it safely. Unplug the microscope and return it to its original location after replacing the protective cover on it to ensure it does not get contaminated or damaged. Fluorescence microscopy can be used for lots of different things. In this first image right here, we see human cells during mitosis. Here, the fluorescent dye is attached to cells that are actively dividing into two new cells. This image helps visualize the process for us. In this second image right here, we see the visualization of the plant cell membrane. This image illustrates the structure of a plant cell and shows the different compartments within. The third one shows a picture of tumor cells. In this image, the fluorescent dye is used on tumor cells, and these can be studied to help find a cure for cancer. And in the last one, we see the nuclei of the cells, right here. The dots in the image are stained with blue fluorescent dye and show the nuclei, or the brains, of the cells. Thank you for listening and following along. I hope you were able to learn a little bit about the interesting technique of fluorescence microscopy.